Good afternoon, ladies and gentlemen, and welcome to those of you viewing online. If you'd like to tweet today, the hashtag is OMS10, but please obviously make sure your devices are on silent. And just to let you know that we do now have guest Wi-Fi here. It's OMS_guest, if you haven't already discovered it. We're very lucky today to have Adrian Hill speaking. He's one of the directors of our vaccines programme. He's also director of the Jenner Institute, professor of human genetics here at the University of Oxford, and a fellow of Magdalen College. He is going to speak today about vaccines and the work that our vaccines team do, and with a particular focus on Ebola, because in 2014, Adrian's group led the first clinical trial of a, I don't know how to pronounce it, monovalent Ebola virus vaccine aimed at targeting the outbreak strain of Ebola in West Africa. So I won't make any further introductions. Please, Adrian.
So thank you very much for that warm welcome and introduction, and congratulations to the Martin School on the 10th anniversary. It's been an exciting year for many of us involved in vaccines, and what I'm going to do, despite this rather daunting title about immune responses and so on, is talk very broadly about vaccinology and vaccines. You'll have gathered we're pretty keen on vaccines. I think they do a lot of good. And I'll talk in particular about one infectious disease, but then try and broaden the discussion to some important areas at the end that I think you might be interested in. So, as you heard, we're part of the Martin Vaccines Programme, which has been generously supported by the school. We're also up the hill at the Jenner Institute on the Old Road campus. We have an excellent clinical trials facility there, and we link very strongly to a veterinary vaccine group at Pirbright, in fact the UK's largest veterinary vaccine activity, supported by the BBSRC. So it's a deliberately broad perspective on vaccine design and development. So vaccinology is the name we give to the science underlying what I think I can truly claim is the most cost-effective medical intervention ever deployed. There is global reach. Every day, in every country in the world, most of the babies and infants are vaccinated. These days, not with a few vaccines, as it used to be, not with 4 or 5 or 6, but in many of the poorest countries in the world, with up to 10 vaccines that are life-saving. And that's a huge logistic achievement and very, very important for global public health. And being in the UK, we should remember that vaccines originally are a British invention, which saved millions of lives. And you know the story, many of you. I will remind you of it in a moment. But it's important to remember that even though I'll be talking about infectious disease vaccines, the scope of vaccination is becoming broader all the time. There are vaccines in development against cancer, a very active field of immunotherapy. There are vaccines that have been in clinical trials and are being developed for Alzheimer's disease, for autoimmune diseases like psoriasis, for reducing your blood cholesterol. And it's a hugely exciting time because the technology is so widely applicable. And as we know more and more about immune responses and what other technologies like monoclonal antibodies can do in the healthcare arena, then vaccination becomes ever more important. So this is Edward, who was a country GP in Gloucestershire, in the town, it's a little smaller now, of Berkeley. And famously, he vaccinated his gardener's son. Now, he wasn't we're pretty sure he wasn't the first person to take some cowpox from a cow and inject it into another person to try and prevent smallpox. But he was the first person to demonstrate that that worked and to publish his results. And for those of you who measure achievement in academia in terms of impact, it doesn't get much better than going from 1796 to 1800, and your experiment on one individual has led to Napoleon, who of course was at war with Britain at the time, demanding vaccine supplies from the UK, Jefferson being vaccinated in the United States, and the Empress of Russia insisting that Russia be supplied with a vaccine within 4 years. That does give us pause these days. It is a very translational activity. We're trying to achieve a result. We're trying to produce something in vaccinology that will be useful. And I would argue for human vaccines that really the only important test of human vaccines is in humans. Despite the fact that most of the research money that goes on vaccine design and development ends up testing vaccines in other species, usually in mice, sometimes in monkeys, but clinical testing is really what this field should be about. Interestingly, unlike drug trials where you often need hundreds or thousands of patients, very often you can test vaccines in very small numbers of individuals and get an initial read on whether they're doing anything. This is the whole field, the rapidly enlarging field of human microbial infections, where you deliberately and ethically, at least so our ethical committees tell us, we can infect people with, say, malaria parasites, which of course are drug treatable, to test whether a vaccine is working. And remarkably, that invention, if you like, also goes back to Jenner, who used a tiny dose of smallpox vaccine to test whether his immunization with cowpox was protective. And because no lesion was produced by the tiny dose of smallpox, he was able to declare that his vaccination had worked, and he was correct. It's also an excitingly translational field. There aren't many DPhil projects that you can do where you actually make something that is your idea with your own hands that can then translate into clinical testing in the few years of your DPhil, even if you don't end up spending 5 or 7 years doing your DPhil. And that certainly happened in our institute. So it is unusual that you can do this in academia. A lot of the drive for vaccine development comes from the academic and non-profit sector and from biotechs, and less from big pharma, who are interested in late-stage vaccine development and able to provide the substantial funds that requires. But what really excites us is that as well as that utility, there is some fantastic science, and I could argue that vaccinology embraces a greater breadth of scientific activity across the spectrum from structural biology to health economics than almost any other academic activity. So some of the science I won't be talking about is the adjuvants and innate immunity, transcriptomics, analyzing RNA after vaccination, learning what pathways are activated in innate and acquired immunity that might give us a read on what vaccines are actually doing. Structural vaccinology, amazing advances in electron microscopy now, where we can literally see to a few angstroms resolution with structural biologists and cryo-electron microscopists the makeup of vaccines that we are immunizing people with. Genome screening, reverse vaccinology, scouring the genome of pathogens. These days, getting the sequence is not the challenge. It's figuring out which bit to use in a vaccine when you have a new microbe, and all sorts of immunological and even therapeutic interventions for vaccination. And for those of you, I know there are many economists in this school who measure impact quantitatively. The growth in the vaccine industry has been pretty spectacular, probably the fastest growing sector of the global pharma industry over the last 15 years. It was about $5 billion in 2000. It's over $25 billion a year in sales these days, much of that reinvested in R&D. There's improved vaccine access globally. We've been pretty innovative as a community in figuring out ways to provide vaccines to populations who just can't afford them and making sure that there is real health impact. And vaccines, of course, have been, with smallpox famously, and are being, with polio, key to disease eradication. There are only 2 diseases that have been eradicated, smallpox in people, rinderpest in cattle, but polio is looking good, down to the last 2 countries, and malaria is even on the agenda for eradication. Measles will happen before that. And as well, remembering that there's a hugely active and very innovative sector of veterinary vaccination, not just for domestic animals and your pets, but also, more importantly, I would argue, for livestock. We've been worried about food security, immunizing animals against all sorts of diseases, notably foot and mouth disease in this country, which caused a £8 billion outbreak not so long ago, is important. Keeping our antibiotics more effective If we can vaccinate rather than using more antimicrobials, that will be important. Biosafety I'll talk about, and outbreaks. And of course, economic growth. I work mainly on malaria. I can make the argument that it is cost effective to develop a malaria vaccine. But part of that argument is that the economic loss to Africa caused by malaria is not just all the deaths in 500,000 children every year, the hundreds of millions of cases, but the loss of GDP to that continent by malaria being endemic there. So there's a very strong economic argument for more vaccine development. So what we did about this exactly 10 years ago, around about the time the Martin School was starting, was we partnered with this institute in Surrey, the Pirbright Institute, veterinary vaccinologists, to develop innovative vaccines, to partner with industry, because we will never sell a vaccine, we will on-license to big pharma, but really to support the One Health agenda, which, as many of you know, is the idea of medics and veterinarians, unusually, sadly, working closely together to deal with public health challenges. And there are many examples of that, from rabies to Ebola to lots of zoonotic diseases. But because we've had this tradition in our universities where medical schools and veterinary schools are very separate, Oxford doesn't even have a veterinary school, that close working, which would be very productive in a research and development perspective, has been missing. We're trying to correct that. A glimpse of what we do. This is the end of the research, if you like. Most research is preclinical, it's immunological, it's done in designing vaccines and so on. You finally get to the clinic, and this is just an enumeration of the number of vaccine candidates that have gone into clinical trials as new vaccines in Oxford over the last 10 years or so. You can see malaria has been a pretty active program, but exciting TB, hepatitis, pandemic flu, and other vaccines, and even prostate cancer down there at the bottom of the list. That started last week. So without going into too much immunological detail, in principle, making a vaccine these days should be pretty straightforward. We know a lot about immunology. I'm not going to trouble you with most of it, but in most cases we can make a fair stab at guessing the immune response that we need from a vaccine, either from studying people who become immune to the disease, maybe in malaria, or doing in vitro tests and seeing if antibodies will neutralize. In other words, they will block the virus infecting cells and protect cells from infection. The second bit has got a lot easier in the last decade. With the explosion of genomics, it is now hours, or at most days, from the identification of a new pathogen to knowing its genome sequence, and even bioinformatically annotating that and predicting what the proteins are that it expresses. And that's what you want to know if you're going to design a vaccine. So subunit vaccine design has gone from really a very difficult multi-year process to one which you can do bioinformatically in hours, if not days. So the challenges are different. The bottom one has become center stage. How do you generate the immune response that you really need with a safe vaccine that is going to be safe not just in the first 20, 50 people you vaccinate, but in millions of people, followed for a decade or more to prove the safety of your vaccine so it can be licensed. And of course, there's a lot to the immune system. Most of our vaccines work in one way. They induce antibodies. These antibodies come in many flavors, but immunoglobulin G is the most important. It's the most persistent, and we spend a lot of our time measuring by an ELISA assay the magnitude of antibodies induced by a new vaccine. So of the 27 licensed vaccines, probably 26 of those work mainly by inducing antibodies. That doesn't tell you that cellular immunity isn't important, but it does tell you we're not terribly good at inducing it by vaccination, and that's a pity, because there are lots of targets like HIV, tuberculosis, malaria, and cancer, where a stronger way of stimulating the immune system, larger numbers of these CD8 T cells, would be hugely important and might be transformative, as it is beginning to look as if that might happen in immuno-oncology just in the last year. So rather than go into particular technologies and so on, what I'm going to do is tell you a story about what happened to us 15, 16 months ago, while our team working actively on malaria, planning new trials, testing new vaccines, and so on, got 2 telephone calls on the same morning in early August of last year. One was from the World Health Organization asking how quickly we could do a clinical trial of a vaccine that had never been in people before. And the other from the Wellcome Trust, very helpfully offering to fund that trial if we wrote a suitable, high-quality grant application. So you know the context. Filoviruses have been known for many decades. Ebola itself was first identified in 1976. Peter Piot, now at the London School of Hygiene, was centrally involved in that at the time. And there was an outbreak in the Congo. There have been many more outbreaks in the Congo in the last 40 years. At least 20. There have been other outbreaks in other parts of Africa, particularly in the Sudan. One of the strains is called the Sudan strain. A close relative, Marburg, causes outbreaks as well in Uganda and other African countries. But there had never been an outbreak in West Africa until last year, or probably the end of 2013. The reason people get excited about Ebola, and correctly, is its case fatality rate, the proportion of people who become unwell, who die. There aren't many microbes with a case fatality rate of around about 71%. We know that fairly precisely now, unfortunately, from the scale of the outbreak of last year. So it's a huge danger to healthcare workers or first responders or to burial teams. This personal protective equipment is required. Unfortunately, that was well demonstrated as well by the high fatality rate amongst doctors and nurses in Sierra Leone early on in the outbreak. So just recapping the numbers, over a year, over 11,000 people died, at least 25,000 cases, probably more than that. And of course, there were no vaccines and drugs licensed. And the central theme of this talk that I'll come back to at the end is why there were no vaccines and no drugs licensed, and why it's so difficult to produce those, not technically, but to make a case for investment in this area. The World Health Organization declared Ebola an international public health emergency in early August 2014, convened ethical committees who took the view that even if there weren't any drugs and vaccines, this was so potentially dangerous, not just to West Africa, but to the rest of the planet, that investigational drugs and vaccines should be trialed very quickly, in fact, as quickly as possible, and hence the request to bring teams together to try and see how quickly that could happen. We had a huge advantage, we the international community, in Ebola, because vaccines had literally already been made, mainly in North America, mainly funded by biodefense funders to protect Americans against the potential threat of a bioterrorist attack. So they were making vaccines that were multivalent, would cover Ebola and Marburg, cover both of the main strains of Ebola, and very luckily they had been manufactured and were waiting to be tested, but unfortunately had never been given to a human being. The two main ones that I'll talk about were the Chimpanzee Adenovirus vector and the Vesicular Stomatitis virus vector. We knew a lot about chimp adenos. We had done most of the clinical trials anywhere at the time in Oxford. We'd just about heard of VSV, and it had been given for any disease to 2 human beings who were lab workers who had pricked their finger while researching Ebola in a lab. Probably not infected, but they were vaccinated in any case. So we knew a fair amount about chimp adeno, even though it's an investigational vaccine technology, and very little about vesicular stomatitis virus. And this is to remind me, of course, that the reason that Ebola keeps popping up in Central Africa is that there is a reservoir in bats, and bats probably infect other primates, and people eat bushmeat, and that's one of the possible routes of transmission. So this is why things changed fairly, or you could see things changing pretty rapidly. In March, WHO were aware that Ebola was there in Guinea, and they, not unreasonably, and they've been criticized a lot for this, but not unreasonably, expected that it would go away. The evidence being that every other Ebola outbreak had gone away, been contained by isolation and basic public health measures in other parts of Africa. What was so different about West Africa and why it exploded is still not very clear. It's probably to do with socio-cultural factors rather than anything special in this viral sequence. Burial practices in Guinea certainly contributed, where people culturally have close contact with bodies as they are buried and transmit from one to the other. Stopping that was not easy. But it spread. There are very porous borders between Guinea, Sierra Leone, and Liberia. The green is Sierra Leone, blue is Liberia, and by the end of September, people were beginning to panic. By October, lots of people were really worried that this was not going to turn around. Luckily it did, around about the end of October. It's not right to say we don't know why. Massive efforts were made, huge amounts of money were poured into those countries. The army moved in, built hospitals, Ebola treatment centers, massive resources, but it was still very difficult in October to know if that was going to work. And I think we were just in time, really. I've talked about One Health. It's worth mentioning in passing that people had been developing and testing vaccines, not for humans, but in chimpanzees, in zoos in North America. Because if you come across a troop of dead chimpanzees in Central Africa, chances are they may have died of Ebola. So this is well known by conservationists to be an issue for other higher primates in Central Africa. So what do these vaccines look like? They're not standard vaccines. Old-fashioned vaccines are usually the microbe killed or attenuated in some way, and you're using the whole virus, and you're trying to mimic the natural immune response and gain immune protection in that way. We don't make so many vaccines that way anymore, possibly because the vaccines that work that way have been made and they're on the market. It's a fairly straightforward technology. These days we're left with the more difficult vaccines like HIV, like TB, like malaria, where that approach does not work. It does a bit for TB. You've got the BCG vaccine, not a fantastic vaccine. But most modern vaccine technology is starting with the genome, as shown on this slide here. Ebola, helpfully, is a tiny genome. It's got 7 genes. Malaria, for example, has got 5,000. And everyone was pretty well agreed on what the best gene of those 7 is to try and make a vaccine with. It's fairly simple. It's the gene that encodes that protein coating the surface of the virus. So if you're going to bind an antibody to the virus, that's an obvious place to start. And the glycoprotein is a sensible size. You can easily put it into an old-fashioned DNA vaccine or a modern adenoviral vector. And that was what a lot of groups did over the last year. But as I say, this particular chimpanzee adenovirus vaccine had already been tested by a group at the National Institutes of Health in North America, and another vaccine had been tested in Canada and shown to work, the Vesicular Stomatitis virus. The same sort of idea. You have a virus here with that short genome, and you replace its own glycoprotein gene encoding the coat of the virus with one from Ebola, and use that as an immunogen. It's a bit unusual. It's of the rabies virus family. We don't have other vaccines like that, except rabies vaccine itself. Was effective in testing in monkeys in America. It is replication competent, which means it has more side effects than viruses that don't replicate, and was developed by a tiny biotech, New Link Genetics, who licensed it on to WHO and Merck during the outbreak. And really this type of technology, which we've been very keen on in Oxford, viral vectors, where you take genes out of the pathogen, put them into safe, attenuated, usually non-replicating viral vectors, really had its heyday last year because of the 7 Ebola vaccines that went into the clinic in a huge rush. 6 of those were viral vectors. I'll talk in some detail about 3 of them actually. And finally, in summer, some data emerged on a traditional protein-based vaccine, which of course needs an adjuvant, as most protein vaccines do. So this really highlighted the fact you can make these vaccine vectors rather quickly and test them fairly efficiently as well. So let's focus on the glycoprotein. This is the vaccine, but in a viral vector. We know about its structure. Crystallographers have crystallized it. We know that there are various points that are susceptible to attack, partly from work with monoclonal antibodies. And some of you will remember ZMapp, which was the great hope in the early days of the outbreak, because it is a monoclonal, it's a mixture of 3 monoclonal antibodies to this glycoprotein of Ebola, and was again developed for biodefense purposes. But tragically, there were only a handful of doses available, so few that we couldn't even figure out with the small number of doses available whether it was actually working or not, and we still don't really know that. I guess it is, but we're not sure. We do know that the vaccines work, at least in monkeys, because in high security, high containment labs, you're allowed to not only vaccinate these animals, but to challenge them with Ebola to see if the vaccine works. So here's a very simple experiment. Top left is the design. You give a single dose of the vaccine, and 4 weeks later, you measure the immune response. The other panels show that the antibody response is pretty impressive with a single dose of this Chimpanzee Adenovirus 3 vaccine. EBO-Z is Ebola of the Zaire strain, and that was the strain we knew was in West Africa. And what this technology is particularly good at doing is inducing cellular immunity, in other words, T cells that can kill virally infected cells. And you measure those by flow cytometry, and what this slide shows you is that there are quite a lot of both CD4s and particularly CD8 T cells, which are potentially protective against Ebola. Most importantly, after a single dose of that vaccine, whatever about the immunology, if you challenge the animals with a whopping dose of Ebola virus, 1,000 infectious units, they're all protected. All should be qualified by the fact that it's typically, in this group it's 4 animals, so these experiments are so expensive, a typical group size is 4 or 5. That's an older strain of human adenovirus. That's the vaccine that we were aiming to test in the clinic, CHAD3. If you wait and see how long the protection lasts, it's not so good. At 10 months after vaccination, it's down to around about 50%. But if you give a second dose of vaccine, if you give the same vaccine, it doesn't do anything because of immunity to the vector. If you give a different vector called MVA, modified virus ankara, that we use a lot here in Oxford, it boosts the level of protection back up to 100%. So that was very, very encouraging. Also, for those of you interested in the immune mechanism, as we are, if you deplete CD8 T cells, those killer T cells, in these animals with a monoclonal antibody, and then challenge them, they've lost most of their protection. So 4 out of 5 animals were infected after depleting the CD8s, no depletion, they're all fine. So we had some evidence on the magnitude of the immune response that was associated with protection in monkeys, and what type was relevant. So this was the plan in early August. Use that chimp adeno-vector, which had been safely used first here in Oxford for hepatitis C vaccination by Eleanor Barnes, Paul Klinneman, and others. WHO, together with us, Wellcome Trust doing the funding, GSK had bought Acaris, which was the little biotech we'd collaborated with in Italy for many years on malaria vaccines. They had developed some of this chimp adeno technology. GSK now owned it. Wasn't why they bought the company, but they did own it. And NIH, who of course had done the monkey studies and were also planning clinical trials themselves. We accelerated all of that, tried to get the vaccine into the clinic as quickly as possible, not just in the UK, but in West Africa, because after all, that's what WHO really wanted to know. Have you a vaccine that would be safe in West Africa for testing? 140 was picked as a reasonable number of people to show safety in before going into thousands in West Africa. You can argue about that. I think it's a fair enough number. And our deadline was the end of the year to have those data, not just on safety, but on immunogenicity, to make the case that this was worth taking further and spending many, many millions on testing for efficacy. We managed to get going by the middle of September with a monovalent vaccine. That just means it's the strain that matches the Zaire virus. 3 different dose levels assessed, and we of course measured safety and immunogenicity, the immune responses the vaccine make. And we were really trying to get started in West Africa as soon as possible. It's very, very unusual to be allowed to test a vaccine in Africa that has not been in human beings in other countries, usually the country where the vaccine was first developed. So we needed some safety data from the UK before the Bali and Ethical Committee would approve going ahead. This was quick by any standard. Those of you who write grant applications to funding agencies, I know there are some in the audience, will not usually write a grant on the 14th of October, send it in and get an award letter, not just a yes, on the 26th of October. Thank you to the Wellcome Trust. Vaccine was filled in North America, especially with the relevant strain. We don't usually go to the UK regulator and get an answer in 4 days, which happened on this occasion. They just put everything to the top of the pile and said they were going to review it. They were encouraged, I understand, by the government. The Ethics Committee met specially and approved it essentially at that special meeting. So the vaccine was shipped by the 11th of September. The lawyers did delay us a few days, signing the last bits of paper, but we got going on the 17th with the first dose, and 2 months later we're finished. 2 weeks later, people were vaccinating in Mali based on these safety data, and this is a fantastic clinical trials unit run by Sam Basso, collaborating mainly with the University of Maryland in Baltimore. This was their first vaccinee being immunized. They, if anything, overperformed, and they vaccinated 90 people in some weeks with the same vaccine. We vaccinated the 60 we were planning to do, and then they set up a special trial center in Switzerland to vaccinate people, MSF and so on, who are planning to go to West Africa. We were not allowed to have volunteers who were planning to go to high-risk areas in a first-in-human trial. Safety profile, reassuringly, was just as we had expected from previous use of this sort of vaccine, a chimpanzee adenovirus. 2 fevers out of 60, but generally pretty well tolerated. And then this was a bit more difficult, even controversial. These are the immune responses we saw in people. Remember I told you there were 3 doses, low, medium, and high. Stronger responses with the high dose, which is what we thought would be the best dose. 93% of people responding, that's pretty good for a new vaccine. And this was the antibody level. You can see it on the y-axis there, about 470 in the full dose group, which is good. And the optimists said, well, that's fine, you're done, single dose will do it. The pessimists, and frankly we were amongst them, said, well, sort of, except that when you look at those monkeys who were protected, their average level was 3,700. None of our vaccinees had 3,700 on that scale. So were the people we gave the vaccine to going to be protected? Truth is, nobody knew, and there was some discussion, I can assure you. What we decided to do, being pessimistic and knowing that we could do better, was to give a second dose of a different vaccine, this MVA booster. We had developed this technology here for malaria, hepatitis C, several other diseases, and very luckily, there was an MVA vaccine sitting in Copenhagen, made by a biotech company funded by the US Biodefense Program to make an Ebola vaccine for MVA, and they gave us quite a few doses. We decided to ask half of our vaccinees back, 30 of them, give them a booster with MVA and see if we could get to the level of immune response that we were confident would be protective. As usually, the MVA was well tolerated, both in Oxford and subsequently in Mali. And here are the results on antibodies. This time the scale has changed. There's pre-vaccination, negligible levels, about 10 to the 2. After the adenovirus, that's A plus 28, it's up at around about 500, you saw that earlier. After the MVA, you're up at about 10,000. So well above the 3,700 we'd set ourselves as a goal. So that was reassuring. We were able to tell WHO, we're very confident with that antibody level that these people will be protected, as the macaque monkeys were. And even better, not long later, the team in Mali had compared giving the single dose with giving the single dose plus the boost, and got very much the same results. So in West Africa this works, you get tens of thousands of units of antibody response if you give 2 doses. And remember I told you that cellular immunity we know is important for Ebola from that macaque study, where if you get rid of the CD8 T cells you lose protection. What were we seeing with T cell responses? So measuring CD8 T cells by flow cytometry. Let's look over here at the CD8s, those are the CD4s for the cognoscenti. After a single dose, detectable levels, these are not bad, 0.3, 0.4, 0.03 and 0.04, and then they go up at least 5-fold after the boost with MVA. So we were able to say the cellular immunity is just as good as well. The problem was that this was an outbreak situation, and you need a very rapid immune response to try and control that. So WHO said to us, yeah, that's really nice, but you're waiting up to 8 weeks between the doses. What happens if you gave them faster? And the honest answer we'd never done that. We had a clue from mice, but we'd never done it in humans. So we changed the protocol, went back and did a 1-week interval and a 2-week interval, and to our delight, the CD8 T cell responses were even better with that short interval, so that's helpful. Antibodies not quite as good, but still much better than with a single dose. So we progressed on with this prime boost regime, and have now shown that this gives high level of response, not just acutely, but for well over 6 months, and we think this is probably the best vaccine out there. But there is competition because, to our surprise, another very large company, remember we were doing the chimp adeno with GSK, Johnson & Johnson came in, put their hand up and said, we've got an Ebola vaccine as well. And they had been quietly doing monkey studies for several years, and they had come up with a regime that involved an adenovirus prime and an MVA boost independently. And their adenovirus was Ad26, the human adenovirus, not very different to Chad3. And they also came to Oxford, I think having spotted how quickly you can do trials here. They came to my colleague, Matthew Snape, and to Andrew Pollard, and they started a trial, I think it was literally Christmas Eve or thereabouts. So they were always a few months behind, but they were very determined and still are determined to develop this vaccine. They did it both ways around, with the booster vaccine first and the adenovirus second. That works surprisingly well, and they get very similar safety and immunogenicity results. And Johnson & Johnson are taking this forward. But that was the second vaccine. The third vaccine, of course, was VSV, being developed by Merck, and they had not been idle either. They got started in about early October, first immunization in America. And by early this year, they had vaccinated in Switzerland, Maryland, USA, Lambaréné and Gabon, Kalifi, that's the Oxford University partnered program, Philip Bijon and team in Kalifi, and Hamburg in Germany. This was a bit more complicated. Most people become viremic after vaccination. About a third develop fevers, measurable fever, and there was arthritis, worryingly, in a significant proportion of people in Geneva. So there was a question for a while as to whether this could go forward. The trial was stopped in Geneva, but not at the other sites, and they managed to identify a dose that looked reasonably safe and were given approval to go ahead. So by the end of the year, both the VSV vaccine and the chimp adeno vaccine were ready, in the sense that we had enough safety data, we had enough immunogenicity data to make the case that this may work as a single dose, should definitely work as 2 doses, and went to WHO, and a very large trial was planned in Liberia, supported by the NIH in the USA. Other actors had appeared. CDC were planning a trial in Sierra Leone. Not too much was happening in Guinea, and if you look at the epidemic curve, the incidence was never as high per week in Guinea as in the other countries, but didn't go away really by the 15th of March, which is when this data series ends on the slide. But of course, in Liberia, where the first big trial was planned, by February and March, there wasn't very much disease left, which is great, unless you're testing vaccines. So this was the grand plan around about the 1st of January this year. There was going to be a 9,000 person per group trial in Liberia, Chad3 against VSV vaccine against placebo control. That was pretty controversial. Lots of people who said, look, it's unethical, people are dying all around us. How can you come in with a vaccine, it's finally ready, and you go and give a third of the people nothing, or a placebo? Lots of debate. Regulators are pretty clear about this. They said, if you want your vaccine licensed, we need a properly controlled trial. And the argument that won the day was really that vaccines are great, but sometimes they don't work, and very occasionally they work in the opposite direction. They increase the incidence of disease. So you can't just go and vaccinate everybody, you won't know what's happening. And of course, by the time the trials really got going, the incidence of disease was a lot lower, and that was a less hot argument. In Sierra Leone, they addressed that by doing a different trial design, where you do a step wedge, as it's called. You start vaccinating in one area, go across the area week by week by week by week, and eventually you vaccinated everybody, so you sort of address the ethical issue. But then you compare the people who are vaccinated early to those who are vaccinated late and see what the disease incidence is. I don't really see that as an ethical solution. If you believe in the problem, you're just delaying vaccination in certain areas. And then the trial that's highlighted here, because it's the one that worked and gave us an answer, was actually in Guinea. It was the last trial to be designed. Arguably, it was a new trial design. And many people will tell you it was a trial design never done before for any vaccine development program. People always had 2 groups, a control group, a vaccination group. This is ring vaccination. It's a very simple idea. You're in a rural area, a case is reported, you send the team out, you vaccinate all around that case, typically in the village, or you don't. That's your clinical trial design. Except in this case, it wasn't a no vaccination control group, it was a delayed vaccination control group. And the fourth one you can forget about because it never happened. That was a very ambitious trial that Johnson & Johnson were planning in Sierra Leone with more than 40,000 people the vaccine wasn't ready in time. But of course the great news is that the Guinea trial worked, or at least the vaccine worked. But I should say the trial worked as well, because this was a new ring vaccination trial design, and the results are on this slide. So here's a vaccine that was first tested in October, and the result of the efficacy trial, phase 3, was published in The Lancet in July, way under a year. So it started in March. You vaccinate villages or clusters or rings, as they're called. There were 48 early rings, 42 late rings. The vaccine wasn't given in time to stop people spreading the virus. Around about 3,500, 4,000 in each group. 0 cases in the early clusters who were vaccinated immediately. 16 cases in the late clusters. Headline result of 100% efficacy. Hard to do better than that. Well, you can do better. Your confidence interval can be lower, 75%, so you can conclude that the efficacy is at least 75% on these data, and there are more data, because this was the first decoding of the trial in the middle of July, and the vaccine carried on being used. And that was very informative in all sorts of ways. Firstly, the headline result is you've got a vaccine for Ebola. Maybe a new technology, there may be some issues about safety in some populations. Safety was very good in this rural setting in Guinea. There's a wrinkle that the vaccine doesn't work for the first at least 6 days, probably 10 days. We didn't really have cases from 6 to 10. But not many vaccines work until you've generated an immune response, so that's not much of a criticism. But from day 10 onwards, it was 100% efficacy. So that means beyond the first month, we don't know how long it lasts for. But this was record-breaking. Here was a vaccine going from first in man to phase 3 efficacy in 9 months. Does, I think, provide lessons for other vaccine development programs. A typical vaccine development program these days will take a decade, mainly more. If it's a difficult vaccine, it'll take 30 years. Malaria is above 30 years and counting. A novel trial design was successfully implemented, and the vaccine probably contributed to stamping out the last few cases in those rural areas. There has been a few cases in Liberia in the last week, but it's been gone for 3 weeks in Guinea. The other thing we learned immunologically is that that fairly modest level of antibody response you get by day 10, and that of course was measured in the VSV trials, is enough to protect human beings against Ebola. And that's important not just for VSV, it tells you that the other vaccines I've been talking about that give much stronger immune responses, not 100 but 400 or thousands if you boost, are almost certainly going to be just as protective. So we've probably got 3 Ebola vaccines, at least, that are pretty safe and probably effective. So going back to the debate about how much antibody you needed, the optimists were right. You don't need very much. And of course the difference is almost certainly that when you rub shoulders literally with somebody who's infectious in Guinea, you get a much smaller dose of virus than if you're given 1,000 infectious units as a monkey or to a monkey in containment. So that's all good news. What's happening now? Outbreak termination. So instead of doing a controlled trial, they stopped the control group and started vaccinating around every case, and numbers went down faster. VSV needs to be licensed. You may have noticed that was a phase 3 trial, and I had been talking about phase 1 trials. Well, for the first time ever, to my knowledge, phase 2 was kind of skipped. So people are going back and doing phase 2, which is where you generate enough safety data and enough immunogenicity data to justify your phase 3, really. You still have to do your phase 2 because a regulator will want safety data in thousands of people with lots of blood samples and so on. So that's going on at the moment for all of these vaccines, for the CHAD3 vaccine, for the VSV, and for the Ad26. The second indication that I haven't talked about is pretty important. You want a vaccine that'll put out the outbreak as early as possible. You also want a vaccine that you can go to healthcare workers or first responders in high-risk regions and say, we'd like to vaccinate you, and it will last a while. And that's what we're working on at the moment. We don't actually know how long these vaccines work for. The immunology suggests that it's pretty good for the adenovirus, particularly if boosted by MVA, but you'd like at least a year. You'd like longer if you could. We don't know that for the VSV vaccine yet. And of course, you need a Ebola vaccine for the other strain of Ebola that's occasionally appearing, the Sudan Ebola virus. So what people are doing are comparing the VSV vaccine that worked. The second column there would say the CHAD3 MVA, and looking at a month, looking at 6 months, now looking at a year, and so on, and trying to figure out how long these are likely to work. So how do you license the other vaccines that didn't get into an efficacy trial? There is a route. It's called the animal rule, where you can show efficacy in animals, so you get the same immune response in people, show lots of safety with a big database of safety in humans, and the FDA and the European Medicines Agency will probably license that. I say probably because it's never actually happened. They much prefer to see real efficacy data, but this, I think, will happen in Ebola. And of course, we do need some stockpiles of vaccines to be prepared for the next outbreak, and that's really the end of this story that I will come on to now. The way I put this is we shouldn't have been doing what we were doing. Not because we were incompetent or should have said no to WHO, but the world should have been prepared. We almost had several Ebola vaccines ready. 3 of them at least were made. Hadn't quite got into clinical trials. If we'd had a stockpile of 5,000 doses, a vaccine shown to be safe and immunogenic, what we did in 6 months, you'd have ring vaccinated in March in Guinea, and that would have been the end of the story, and I wouldn't be talking to you today. So we were almost there, but we weren't. So we need to be prepared in future, not rushing to do a very rapid response. And the broader context is it's not just Ebola. There's been SARS, there's been swine flu, there is MERS at the moment. In fact, there are 15 known knowns, as Mr. Cheney might put it. There are potentially lots of unknowns. SARS was totally unknown when it first emerged some years ago. And the bottom line is the worry. We have no licensed human vaccine for any of these. Ebola isn't even licensed yet, even though we have a vaccine that works. There's one other that's hopeful. Enterovirus 71 has gone through phase 3. We may have a pandemic flu vaccine if the right strain comes along. If the wrong strain comes along, we don't have a vaccine. But for the rest, for MERS, Rift Valley fever, chikungunya, Nipah, you need other public health measures. And the irony, I think, and Ebola is a great example, is these are not difficult vaccines to make, I would bet. Ebola worked first time. If you do mouse immunization experiments, you get antibodies that neutralize the virus. I'll show you a couple of examples in a moment. So these are not on the scale of difficulty of HIV or malaria or cancer. I think these are doable. It's just that nobody's ever done them. And of course, the question is why? Will we need these vaccines? Almost certainly. There are more people, especially in Africa. Cities are bigger. Monrovia and Freetown are much bigger than they were 10 or 20 years ago. Much more long-distance travel, especially in Africa. Flight route numbers have increased in Africa more than in the rest of the world. These viruses are being transported around somehow, and there are lots lurking in bats and caves and elsewhere, and more will appear. So the problem, of course, in a word, is money. Why would any company, even if it's very big, spend $100, $200 million developing over 5 or 10 years a vaccine that is targeted at a poor area of rural Congo, where outbreaks might occur again, or might not, where they might well be controlled by good public health measures as countries become richer, and you might never need the vaccine? Well, of course, they didn't. The vaccines that were developed on a biodefense program started after 9/11 to protect bioterrorist outrages in North America. So there's a business case failure, if you like. There's no business case for doing this. So what I've been proposing is that you could do it less expensively, and a lot less expensively, if you decided we don't have to license the vaccine to get it used. So you could do what we've done for Ebola. We know how much that cost. It was tens of millions, maybe $20 million across the several programs, all in a year. And you ended up with small batches of 3 vaccines. You know they're safe. You know they produce a good immune response. At least one works. The others do as well. That was pretty cheap. Could you do the same and make a small stockpile for all the others, for MERS and so on? Yes, I think you could. And that's going to cost you tens of millions rather than hundreds of millions. You don't get a license, but think about it. If you've got a vaccine that probably works and there's an outbreak in, say, Uganda, would Uganda want to use that vaccine? Of course they would. It's safe, it's immunogenic, it'll probably work. And as a bonus, you find out if it works, because the only way to really find out is to use it in an outbreak setting. I would argue you need stockpiles held in affected regions. That's a single manufacturing batch I'm proposing, 10 to 50,000 doses. There are such things as emergency use approvals. They were used in West Africa for Ebola, and you can measure efficacy if you do the right trial design. And this is absolutely no news at all to veterinary vaccinologists. In fact, in this country, we have pretty good stockpiles of foot and mouth disease virus vaccine in case there's another foot and mouth outbreak. They're down in Surrey. So the idea is not new, but implementing it for outbreak pathogens, I would argue, is urgently needed. What about these other pathogens? Well, here's some examples from work we've done here in Oxford. This is George Rimway, who's a Kenyan vet who's been with us for several years. He's used the same technology, put a Rift Valley fever antigen into these viral vectors. And rather helpfully, this is not well, it's not helpful actually, but this is not just a human pathogen, it's a pathogen of goats, sheep, and cattle. That means George could do challenge studies actually in Kenya with those three. He not just got really good neutralizing antibodies, he's got 100% efficacy in cattle, sheep, goats, and has vaccinated camels. Yet he hasn't actually infected them, but I'm sure he's going to do that fairly soon. But it's looking great. Similarly, for MERS, we have made constructs. That's fairly straightforward and routine. We get good antibody levels. The efficacy trials are not difficult. Maybe we should do that fairly quickly. And rather helpfully, the UK government was pretty quick off the mark on doing this. The World Bank has been putting up its hand saying we need a special fund for outbreak pathogen vaccines. Of course we do. We just need somebody to lead on it. David Cameron announced 20 million at the G7 summit in Berlin this year. Now, that's not going to make you several vaccines, but yesterday, actually to our surprise and delight, 20 million became 120 million in the autumn statement. So that's progress. And other parties are very interested. The Gates Foundation, the Wellcome Trust, and others will likely contribute. And, you know, this is a global problem. We don't need 5 people to make an effective vaccine, but we do need 1 for each of those outbreak pathogens. So, in summary, modern approaches can generate effective vaccines for most infectious pathogens. The ones you hear about are the problematic ones. We've made vaccines for most useful pathogens. HIV, RSV, malaria, TB are the exceptions. They're uniquely different for rather special, difficult for rather special reasons. You can do clinical trials faster if everybody helps and allows you. We've seen that. Maybe we should be doing that. The reason it costs hundreds of millions, a lot of that's the delay and the teams waiting for approvals. The response in Ebola has been unprecedented. I think sets a great example for how we might do other things faster. But then worryingly, many viral threats against which we have no human vaccines are out there. Those are the known knowns. There are also unknown ones. And we need a new strategy to develop the vaccines for these vaccines for which the business case is clearly weak. So just to mention that I've been reporting for a huge number of people who stood up in August and decided they would stop doing what they're normally doing and help out with Ebola. And it's not just all the people who went to West Africa who were fantastic. It's those who did trials around the world, who turned over their specialized facilities, whether you're doing macaque studies or clinical trials or whatever, to help deal with this emergency. And happily, the world has ended up with an Ebola vaccine. So I will leave it there and be happy to take questions. Thank you.
Thank you, Adrian. That was fantastic. A really good insight. We'll take questions. Obviously, this, like I said, this is being live webcast and recorded, so if you don't want your voice or face on film, get a mate to ask the question for you. At the back first.
Thank you for your very informative talk. In terms of the principle that prevention is better than cure, do you advocate that populations globally and locally are, as far as is possible, helped to remain at what might be a more helpful, lowest possible level? Secondly, in terms of human behaviour, is there some means of helping people to either not eat bushmeat or eat it in a safer manner, and to have a safer manner of funeral practice, burial practice? Thank you.
Yeah, really good questions. There's an ongoing discussion about where you want to be with Ebola, say in 2 or 3 years' time, assuming you have licensed vaccines. These can be manufactured at a sensible price. So what would you actually do? Would you vaccinate all of those 3 countries? Yes, that would be helpful. We'd like to know how long the vaccine works for first. We're working on that. If it only works for a year, that's unlikely to be cost effective, remembering there's only been 1 outbreak in West Africa. There are other countries where outbreaks occur more frequently. For example, in the Congo, I didn't mention that during all the panic about West Africa, there was another outbreak in the Congo. 49 people died. About 100 people were unwell. And it was controlled, as it's usually controlled in the Congo, by simple isolation. So there are probably more outbreaks going to happen in Central Africa than West Africa. The strategy I like most at the moment, and there's a lot of discussion, is that you would vaccinate routinely healthcare workers, first responders, burial teams, and just offer that as part of people's job. So, we do a great job vaccinating babies. The taxpayer mainly pays for that through Gavi, through UNICEF. And it's hugely cost effective and saves millions of lives. If you think about cost effectiveness of an Ebola vaccine, it's actually quite tough to make the case because you don't know when there's going to be another outbreak and you've got to estimate how much it will cost. So if you pitched up 3 years ago and said, I want to vaccinate people with an Ebola vaccine, here it is, it would have been turned down on cost effectiveness grounds. But it's actually not cost effectiveness, it's international security. So the cost of the outbreak has been estimated at around 5 billion. If you add up the cost not just in West Africa but in other countries, we spent a lot of money being prepared, etc., and so on. So a case is being made to say there needs to be a different agency, and this is not my idea, this is Dr. Kim at the World Bank, who's a medic as well as an economist, saying that there needs to be a different strategy for prioritizing vaccination of, say, healthcare workers or people who are likely to be first exposed, or maybe butchers dealing with bushmeat, if that's implementable. And the suite of vaccines you would give those individuals is different to those you'd give every baby in Central Africa. And the benefit is different. It's not just reducing the risk in those individuals, it's reducing the response required and the cost of that response for the rest of the world every time there's an outbreak. And we've been having significant outbreaks every 1 or 2 years over the last decade, and if population growth and mobility increases, that's likely to increase as well. So I think a lot of work needs to be done on getting vaccines that are suitable for dealing with these outbreak pathogens, finding the right population to vaccinate, introducing that vaccine in a way that's acceptable to communities that are not used to adult vaccination or teenage vaccination, and moving forward in that way.
Who's next? Just in the middle here.
Is Ebola a disease that may recur? I think there was the case of a healthcare worker who may have had a recurrence, and if so, is there any value in vaccinating people who've been infected already?
Yeah, really good question. So the answer is yes. It is a virus that hides in what medicine calls immunoprivileged sites. Most notably in males, in semen, so it's sexually transmissible. There have been several cases recorded of sexual transmission from males to females. One has been published in the New England Journal of Medicine. It also hides out in the back of the eye, where immunity is not as strong. There's been one unique case in this country that had a lot of attention and caused a lot of concern. So the Glaswegian nurse who was flown back in about January, was in the high containment healthcare facility in the Royal Free, was discharged well around about the end of January, and at the beginning of October had a recurrence. We don't know all the details, but we're told she was viremic from Ebola again. She was back in the Royal Free very, very quickly, quite seriously ill, had meningitis, and has happily now recovered. That's very unusual. There are 11,000 people walking around in West Africa. There aren't reports of meningitis due to Ebola months later. So quite what's different in her case is not clear, but I think the biggest risk is sexual transmission. The cases that are popping up, like the 3 family cases in Liberia in the last week, where are they coming from? We don't know. So a lot of work is going on to trace these. Of course, you can sequence the virus and identify exactly what it is and likely who it came from. But Ebola won't be eradicated. There'll be reservoirs in bats, but it's not even clear it will be eradicated from the human population. There are carriers. The carriage reduces over time, so the rates are very high in the first 3 months in semen. By 9 months, it's much lower. Those men are being followed, but this is complicated. And your question was, if you vaccinated those people, would you get rid of the carriage? It's a really interesting question. I think it should probably be done. Unfortunately, I think the answer is no, it won't work, because they have really strong immune responses. So I was showing you numbers on the magnitude of the immune response. Even in our best responders, if you compare those to people who've had real Ebola and measure their antibody levels, the vaccine isn't as immunogenic for antibodies as a dose of Ebola disease.
Any more questions? Oh, you've obviously answered them so thoroughly, Adrian. Thank you for that. Just before we thank Adrian, I just want to flag up our final seminar of the term. It's same time next Thursday, and it's Tim Palmer, one of our climate scientists. So do come along to that. COP will have started by then, so the world will be gathered in Paris to discuss how we tackle climate change. So Tim will have some very interesting insights on that. So all that remains is for us to give Adrian a warm round of applause for a great overview.